Hi, I'm Andrea Patterson, and I'm currently a third-year graduate student in the lab of Dr. Craig Bird at The Ohio State University. I would like to take this time to give you the highlights of our recent molecular endocrinology paper, Sustained Reprogramming of the Estrogen Response Following Chronic Exposure to Endocrine Disruptors. To address growing health concerns that compounds known as endocrine disruptors can mimic endogenous hormones like estrogen and alter estrogen receptor signaling, our lab developed a chronic treatment model in the estrogen-dependent breast cancer cell lines MCF7 and T47D. Endocrine disruptors, bisphenol A, an industrial estrogen, and genistein, a phytoestrogen, were administered for over a year in culture in the presence of physiological hormones in order to mimic human exposures. Seen here, MCF7F representing the control cell line passaged in parallel, and MCF7B and MCF7G representing the chronically treated cell lines with bisphenol A and genistein respectively. We found through microarray analysis that most genes with altered expression were more highly expressed following chronic exposure to bisphenol A and genistein, shown here with many more genes upregulated in our chronically treated lines and fewer downregulated. However, estrogen-responsive genes were significantly downregulated due to this chronic exposure, seen here with over 200 genes upregulated in the control line in response to estrogen, compared to the 100 or less genes upregulated in the chronically treated cell lines. Closer examination of known estrogen-responsive genes revealed that chronic EDC exposure initiates compound-specific transcriptional reprogramming. Specifically, estrogen receptor target genes showed decreased expression, as shown here with the EGR3 gene target with significant repression, and reduced receptor recruitment to its response elements. We show that these alterations in estrogen receptor signaling were epigenetic through observed loss of H3K4 monomethylation, a mark for enhancer elements, and decreased DNA accessibility. These data demonstrate the chromatin at response elements is more compacted and less accessible to the receptor. Finally, these alterations in estrogen receptor signaling were found to be persistent, and that upon removal of these compounds from culture for several passages, the defects in gene expression, signaling, and chromatin landscape remained mostly unchanged from the previously observed altered state. Our work demonstrates the potential of chronic exposure to endocrine-disrupting compounds to permanently alter hormonal signaling, validating growing concerns that prolonged exposure to these compounds can result in lifetime health consequences. We hope we have convinced you of this upon reading our work and invite you to read our paper for more details. We would also like to acknowledge support from the National Institute of Environmental Health Sciences, the Ohio State Comprehensive Cancer Center, and our home program, Solid Tumor Biology. Thank you for watching.